We're seeing that youth do have a more aggressive disease, develop complications far earlier than we would observe in adults, and we clearly need to pay attention to this and find novel ways to approach this disease. The RISE Consortium performed 3 different protocols as feasibility studies to look if we could find approaches that will preserve beta cell function in individuals at high risk of getting diabetes or those with recently diagnosed diabetes. At the American Diabetes Association meetings today, we presented the results of the adult medication study and contrasted those with the data that we had obtained in youth. In these 2 protocols, one in adults, we used 3 different active interventions, metformin alone, or glargine followed by metformin, or liraglutide plus metformin with a comparative placebo arm, each used for 12 months and then a 3-month washout. And in the youth, we had a metformin alone arm or a glargine for 3 months followed by metformin arms with treatment ending at 12 months and a 3-month washout, which allows for a comparison between the findings in adults and the findings in youth.
Dr. Kahn, we heard the results of the RISE study, very, very interesting. The study, because it had both children and adults, I think is challenging the way we think about impaired glucose tolerance and type 2 diabetes. Dr. Taylor, do you have a question for Dr. Kahn?
I just wonder if you'd comment on how that might impinge upon clinical practice for the initial drugs used in type 2 diabetes, firstly for adults and perhaps separately for children.
I would suggest that we've learned a lot from these 2 studies for both initiation of therapy in adults and in youth. And in the comparison, we looked at the effects of glargine followed by metformin and metformin alone. And in adults found that there was an improvement in certain aspects of beta cell function measured as responses to intravenous glucose and other secretagogues. And these benefits were present after 12 months of therapy. Unfortunately, what we also witnessed is that when you stop the therapy, after 3 months, everything returns back to baseline. In contrast to adults, where with glargine for 3 months followed by metformin, or metformin alone for 12 months, where we saw an improvement while on therapy, which was lost during the washout, in youth, we failed to see such an improvement, and at the end of the washout, it was very clear that they had progressed even further, so that their beta cell function was significantly worse than they were at the time of initiation of the study. Now, this doesn't, in my mind, say to me that we should not be using these medications in kids. Absolutely not. We must use these medications in youth, and I should also add, we did not have a placebo arm in the study in youth, so we don't know really whether the progression we had seen would have been worse if there had been no intervention.
I think one of the very interesting things about this study is it had both children and adults, but it was surprising that the children or the adolescents and adults with type 2 diabetes had a different response. And I wondered what your thoughts were about whether they have 2 different diseases or why they might have responded differently.
Is the disease different in youth and adults in terms of its pathogenesis? Or is this really the same disease that is being accentuated in some form by the fact that adolescents have a variety of other things that are changing at the time, whether it be sex steroids and puberty, whether it be growth hormone and IGF-1, we don't know. It creates a whole series of questions that will be critical for us as scientists to address in the future. And in that process, potentially also identify new targets for therapies that would hopefully allow us to slow or even arrest the disease in youth, and then potentially also translate those same observations and therapies into adults.
Could you just describe for us how you see insights into the mechanisms of beta cell failure and what's actually happening as a result of prescribing these agents?
Yes, the beta cell puts out more insulin and seems to be doing better under those circumstances. But unfortunately, when we withdrew the therapy and waited 3 months and retested individuals, all those potential benefits had disappeared. So what that means in the long run in terms of mechanism for disease, I don't know, but we made also some interesting observations when we look at the different measures of beta cell function that we were testing using the intravenous tests that suggest that the beta cell may have a finite capacity to secrete insulin, and that what we were seeing with the liraglutide was it was able to push the beta cell to its maximum to secrete. And while it could do that, it at the same time wasn't doing anything in terms of preserving the cell. In contrast, in the youth, as we saw, they demonstrated no real benefit of therapy during the 12 months of the active intervention, which says to me that underlying the changes that we observed is a difference in the pathophysiology that drives these youth to failure more rapidly. That we're seeing that youth do have a more aggressive disease, develop complications far earlier than we would observe in adults, that suggests that this disease in them is really quite severe, progresses rapidly, and we clearly need to pay attention to this and find novel ways to approach this disease, both in terms of preventing beta cell progression and clearly treating the potential complications.
ADA TV is brought to you from the American Diabetes Association's 79th Scientific Sessions. For more from the meeting, make sure to click these links and subscribe for much more from the world of medicine.